Hi, this is Miss Linton. Say hi.
Hi.
And this is Honors Bio Review for Unit 6 exam. Now I just want to remind you, because we have the rally tomorrow, to bring your headphones, right? Because you're going to start your hyperdoc. So text your friends. No, it's not a rally. It's art. Art, right? Fine Arts Assembly. I can't remember anything. Okay, Fine Arts. So bring your headphones because we pushed the test back until Monday, right? But we wanted to do our review today. All right, so this is your central dogma unit. What is that a picture of? DNA. You should know a lot of things already from there, right? We could talk about DNA structure from this diagram. What shape is it? Who came up with that? What? I'm sorry, I couldn't hear you. Watson and Crick. They used information from some other scientists. Do you remember what those scientists were? Chargaff. Perfect. Let's talk about Chargaff first. Chargaff, when he did his research, he said, however many adenines you have, you'll have the same number as thymines, and however many guanines, you'll have the same number as, and then your AT-GC ratios are very specific to your species. But A's and T's will be equal, and G's and C's will be equal. Who else did Watson and Crick use information from besides Chargaff? Franklin. Yes, Rosalind Franklin, who kind of went, remember? And so, and there was maybe some thievery, right? Okay. And so what she did, and Maurice Wilkins, they sent what through DNA? X-rays, through crystallized DNA, and it came up with a pattern, right? And that pattern told us that whatever DNA was, it had to be in the shape of a helix of some sort, and that it was the same diameter across, and there were repeating units. And we know that because you have a purine always connected to a pyrimidine, right? And so it keeps that diameter the same. This model is not very good. Why? Because it shows like the two sides of the ladder are exactly the same, but you know it'll move, right? The diameter will stay the same, but you have a purine with a pyrimidine. A purine has how many rings? 2, and a pyrimidine has 1. So it fails in explaining that part. You do know what holds the two chains together. I'm glad they have a little bit of a gap right there. And that gap would be what kind of bonds? Hydrogen bonds. How many between guanine and cytosine? 3. How many between adenine and thymine? 2. Excellent. Good way to start. All right, so chapter 24. This is a review I put together to get ready for the final last year. So it's including some of the biotechnology that we are skipping over and doing later. So I'll skip over it in this review as well. Okay, so topics to review in chapter 24. The one you would not need to know for this unit is this right here. We will learn that later, okay? But these are the other topics that we need to know. DNA, we want to know the history of DNA, like some of the key experiments. We've already talked about 3 of them. The structure of DNA and how it gets replicated. Then we want to talk about gene expression, which is transcription, which is how you use DNA to build what? RNA. All kinds of RNA, right? But if we're going to translate it, we're going to need to use that mRNA with tRNA and rRNA in order to build our protein. So that's translation. And then we also talked about mutations, when it doesn't go so well. When there's a mistake either in the DNA or the RNA, and as a result we have proteins that have some issues. Okay? And we talked about frame shift and point mutations. All right, can I keep going? Okay. So we started off by talking about a bacteriophage. A bacteriophage is actually not bacteria at all. It's a what? Virus that infects bacteria. Now, we had 2 scientists, Hershey and Chase, that worked with the bacteriophage. And the purpose of their experiment was to find out what is it that viruses put into bacteria to take over the host cell machinery and get it to make more viruses. And they knew that viruses were composed of 2 components, protein and nucleic acid. Now, they didn't know the structure of a bacteriophage as I have shown you here. They had never seen it. They didn't have the microscopy to see that. But we now know that there is an outer capsid made of protein and then an inner nucleic acid. And it's the nucleic acid that it shoots into the bacterium. But they did not know that then. So they labeled the nucleic acid with an isotope P32 of phosphate, and they labeled the protein with an isotope of sulfur S35. And then they went to see what ends up getting in the bacteria. So the way that works is bacteriophage will land like a lunar module onto bacteria. It'll inject something in, and then to separate the virus from the bacteria, what did they use? A blender, okay? And the virus will, and the lunar landers fall off. Then they take that fluid, okay, pour it into some tubes, centrifuge it. The heaviest parts go to the bottom. Who's going to weigh more, bacteria or virus? Bacteria. Viruses are so small they're not even alive, right? But it's not because they're small, it's because they don't have the characteristics of life. Okay? And it separates the bacteria from the viruses. The bacteria at the bottom, they just need to see what was in there. Was it radioactive phosphorus and nucleic acid, or was it radioactive sulfur and protein? And it was radioactive phosphorus. So they knew, okay, when a bacteriophage overtakes a bacterium, it's using a nucleic acid to do it. And that pointed to that is our code, along with some other experiments as well. Okay, we already talked about this. Who should you think about? What experimenter should you think about when you look at this? Chargaff, good. Okay, and here we can see Rosalind Franklin, and she sent the x-rays through. And here we can see the structure of DNA by Watson and Crick. We know that purines are bound to pyrimidines, and I would know that there are three hydrogen bonds between guanine and cytosine, and 2 between adenine and thymine. We know the replication of DNA. When would that happen in the cell cycle? S stage of interphase, right? This is when, if we go back to my analogy and my modeling, this would be making another whole copy of my cookbook, right? So we know when that happens, all the DNA needs to be replicated. And when that happens, we call it semi-conservative replication, because each old strand of the DNA serves as a template for a new strand to be built off of it. So if we have an adenine here, we're going to bring in a nucleotide. A nucleotide is a base, a sugar, and a phosphate. We're going to bring in a thymine. If it's a guanine, cytosine. Okay, and that is called semi-conservative replication. And these right here would twist up and form sister chromatids held together by a centromere that separate during anaphase. Perfect. Okay, that is DNA replication. Little reminder, DNA and RNA are different. DNA is double-stranded, RNA is single-stranded. DNA will have the base thymine, RNA will have the base uracil. DNA will have the sugar deoxyribose, RNA will have the sugar ribose. Perfect. Okay? So our next step is, now I have 2 cells. Right, I just did mitosis, right? I made a copy of all the DNA. I have 2 cookbooks in each of my homes, yes? Now I want to make a protein. I don't need to copy the whole cookbook to build my pie. I just need to copy out one recipe, because my cookbook can't leave the cupboard, can't leave the nucleus. That copy that I make is called what? mRNA. M stands for messenger. Where did it get the message from? DNA. I will also transcribe rRNA to be part of a ribosome, and tRNA, and what does that stand for? Transfer RNA. What does it transfer? Amino acids. Okay, because those are going to be part of the process of making proteins. So I will transcribe all 3 RNAs, but only one of them will be translated into a protein. Which one gets translated? Yeah, the mRNA. Okay, so when we look here at this picture, transcription is the formation of mRNA. Translation is using that mRNA on a workbench. What's the workbench called? Ribosome. Ribosome is made out of what? RNA and proteins. Okay, and using tRNA to transfer amino acids. I know the sequence of amino acids because the tRNAs have anticodons that are complementary to codons on the mRNA. The codons were complementary to the DNA. So the DNA determined the mRNA. Every 3 bases on the mRNA is called a codon. tRNAs will land at the appropriate place on our workbench, bringing with it amino acids. Hook the amino acids together to build a protein. So who ultimately tells me the sequence of amino acids in my protein? The DNA. DNA determines the mRNA. mRNA determines the proteins, right? The amino acids in your protein. That is referred to as the central dogma. DNA to DNA is replication, DNA to RNA is transcription. Using that RNA to build a protein, we're translating it. We're translating it into a new language. This was nucleic acid language. This is protein language. We translated it into a new language. Transcribe, we kept in the same. RNA and DNA are both what? Nucleic acids. We're just transcribing it. Translation makes us build that protein. Now, one of the things we talked about, when we do transcription, we're making a complementary copy of that DNA. But we may want to modify it. I may want to put a little more sugar in my apple pie, or I may not want any sugar in my apple pie, or I may want to put peaches instead of apples, right? So I can modify the mRNA. The parts that I keep are called what? Exons. The part I cut out are called introns. How do I remember that? Exons are expressed in the protein. Perfect. Now, I need to protect my RNA as well, so I put on a cap of 7-methylguanosine and a tail of 100 to 200 adenines. And that protects either end of the mRNA. Okay? Now, here I am translating. Codons on the mRNA are complementary to anticodons. If my codon on my mRNA says AAA, my anticodon says UUU. What do I look up, the codon or the anticodon? I look up the codon. I would look up the AAA to see what amino acid that tRNA is bringing me. That will tell me what's getting transferred to me. That's the message that I use. So we can see that again in this diagram. Let's call this guy tRNA, here's tRNA1, okay, he's right here, or tRNA 1, 2, 3, 4, 5, okay, tRNA 5, he's got all 5 of these amino acids here. Here's tRNA number 6, he lands. These will get dumped onto the new guy, and this guy will leave. How does the dumping get facilitated? The ribosome shifts and facilitates that peptide bond between the amino acids. And this will continue and continue and continue until I reach a stop codon. Okay, until I reach a stop codon. Okay, then we said, what can cause mistakes? Well, if you mess up the DNA, then therefore your RNA is going to be messed up, and therefore your protein is going to be messed up. So what kind of things will mess it up? Well, we could have made a mistake when we replicated, but that's very rare because there's lots of checks and balances for that. We could be exposed to some chemicals in our environment, mutagens, that may be, did I ever push record? I did. I could be exposed to mutagens because I smoke, let's say, 20 cigarettes a day. You know, just light one right off the other. I don't. Okay, well, let's say I did. I'm exposing myself to those mutagens. Let's say I light one cigarette right off the other while sitting out in the sun without sunscreen, and I pour some chemicals on me, and I'm overweight, and all I eat is fatty food. Okay, I'm setting myself up to have some mistake on my DNA. Another thing that can cause your DNA is transposomes. This is when DNA says, you know what, I'm leaving here, and I am going to jump in here. Okay, those are what kind of genes? Jumping genes. Exactly. So that could change our DNA. An example of jumping genes are Indian corn, because one gene is jumped in the middle of the color or pigmenting gene, which changes the color and gives it this appearance. Once that is in that corn, that's forever until some other mutation takes place, right? So it just keeps accruing those mistakes over time. Now, a really bad mutation are called frame shift mutations, because this screws up all your codons from there on out. And the analogy I gave you that was the 3-letter words. 3 letters just like codons. That's why I use that. So the big fat cat ate the big rat. If we have a deletion and we lose the E, then the first codon is not going to be THE. The first codon is going to be what? THB. And that's where we get, you can say it with me, they get if a attic at a tet have a igor at, right? So now it does not sound anything like what we intended. Every single amino acid is screwed up and our protein is screwed up because we had a frame shift. We're all off by 1. Okay, that is later. Okay, jumping in here. Chapter 25, control of gene expression and cancer. So when we look, first of all, are there any questions on 24? Did I move too quickly, rapidly, things you need me to address? Okay, my smart ones. Let's move on to chapter 25, control of gene expression and cancer. So here are your big ticket items on chapter 25, okay? So what are some characteristics of cancer cells? I would want to know that. That's when things go bad. What is the difference between proto-oncogenes? An example is the RAS, or tumor suppressor genes, and your example there is P53. We said, what are the jobs of all of these things right here? And what are the 2 main causes of cancer? How do you diagnose it? How do you treat it? This is basically all of chapter 25 on one screen. So let's start with this right here, okay? We looked at prokaryotic gene expression, and in prokaryotic gene expression, we have some coding and non-coding sections of DNA. Some non-coding sections mean they don't code for any proteins, they're just binding sites, okay? What is one of those binding sites? Promoter. Who likes to bind to the promoter? RNA polymerase binds to the promoter, okay? That's how it docks in, right? You can't just throw your jacket together like this. You need the zipper to dock in and then brrr, you can read it. So this is where RNA polymerase docks in. It will read the mRNA, okay? What's another non-coding site? The operator. Who likes to bind to the operator? An active repressor. If an active repressor is sitting on the operator, RNA polymerase is unable to attach to the promoter. Okay, because these are your starting off points. There were 2 coding sections of our DNA. Do you remember the 2 coding sections? Regulator gene, if transcribed and translated, makes a repressor. Okay, and whatever structural genes we're trying to control. In the example that we used, these structural genes coded for enzymes, and these enzymes would digest lactose, right? What is lactose digested into? Glucose plus galactose, okay? So they would code for these. I don't need to have these enzymes unless I have what present? Lactose. And lactose would have the ability to remove the repressor from the operator so RNA polymerase could bind to the promoter and transcribe those genes. That was typical of prokaryotic cells. Okay? Now, eukaryotic cells, we looked at levels of control. So it's not just so much about whether we have repressors or not. In fact, in eukaryotes, we generally don't have repressors. It's about transcription factors and activators which assist RNA polymerase to bind to the promoter. But we have all these areas where it can be controlled. Chromatin structure can be controlled because DNA can be tightly wound and put away. What was an example of tightly wound, put away DNA? Bar bodies. The inactive second X in every female cell was a bar body. You're not going to transcribe and translate that. Okay? Transcriptional control, this is where we talked about the Boy Scouts, right, that come along, the transcription factors that help RNA polymerase bind to the promoter. But it might need an enhancer that's bound to an activator and 2 other transcription factors, and now RNA polymerase will chug along. And this right here is the largest point of control, transcriptional control. Post-transcriptional control, we've made our mRNA with its codons. But this is where we're cutting out the what? Introns, capping, tailing, letting it leave the nucleus. Translational control, We are building our protein on a ribosome. Can we do it, or is something getting in our way? And post-translational control is how we wet that protein, fold it into the right shape so it has functionality. So these are all levels of control that you could have for that. Okay, and these were some of the diagrams we looked at. Right, regulator gene, repressor, then we could remove it. And I asked some questions, and if we have time, I'll go back and do those questions with you right here. Okay, this is a picture showing you transcription factor is like this. I don't really like that picture. But this is where you had to have RNA polymerase get some assistance in order to bind onto the promoter. I don't know why I left that one here. When we talked about heterochromatin and euchromatin, heterochromatin is dark staining DNA, and it's inactive because it's tightly coiled up like a bar body. And then euchromatin is the light staining, that's the stuff that's active. So here's an example of a bar body. This is a diagram of post-transcriptional modifications. Cap tail, cutting out introns, being able to leave the nucleus. Here is translational, and then post-translational control, folding that protein. Okay, we then finished up this unit talking about cancer. And we talked about that normal cells and abnormal cells look very different. Their nucleus looks different. The amount of cytoplasm they have looks different. They have an abnormal number of chromosomes. They behave differently. Normally, cells exhibit what we call contact inhibition, right? But abnormal cancerous cells do not. They don't care if it's crowded. They'll build themselves a tumor, right? We said that you need anchorage dependency. You need to be attached to some substratum. But cancerous cells, they don't need that. Something's wrong with their DNA that's causing a problem. They defy the normal regulatory mechanisms that our body has, and they have the ability to spread to other places and defy it all over your body, okay? So we talked about how cancer cells, though, would be contained and would be benign as long as they didn't have access to any blood, because they need the blood for oxygen and nutrients. But angiogenesis is the ability of tumors to be able to call blood vessels to them in order to establish a connection with oxygen and nutrients, and they can expand. Okay, then we broke it down into two basic genes. Okay, remember we said it has to fail in two areas. You have to step on the gas when you're going into an intersection. You have to say, go cell cycle, go cell cycle. So you're racing into the intersection when you shouldn't be. And also, what else has to fail? Your brakes. Your brakes have to fail that would counteract that out of control stepping on the gas. So proto-oncogenes are like generals. When they become rogue and bad, then they will promote the cell cycle even though they haven't received a signal to promote the cell cycle. And our good example of that, do you remember the name of that gene? RAS. Yeah, RAS is like stepping on the gas. And it's in pancreatic cancers, leukemia, lymphoma, that is establishing all of those cancers by saying, go cell cycle, go, go, go. Make more white blood cells. Okay, and so here's an example of RAS. Normally it is only triggered in normal situation by a growth factor binding to a what? Receptor, which then triggers the RAS. But here, this RAS is not getting triggered by a receptor, and it's saying, anyway, go ahead, let's go ahead and make more cells. Now, if everything else was fine in our body, that would be handled. Okay, because it would be handled by what's called tumor suppressor genes. And tumor suppressor genes are like assassins. They say stop. It's like stepping on your brakes. But if those tumor suppressor genes, which the most common one is P53, if he is not there to say stop, and the cell cycle keeps going, 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 if you don't wipe them out, that bad cell, that's when you have cancer. Out of control and no assassin took you out. And we talked about, well, what are the odds of you getting cancer? Well, you could inherit cancer if it's a genetic type of cancer, like the breast cancer gene, right? Or you could put yourself in situations like being without sunscreen and smoking and being overweight and exposing yourself to mutagens, which could also screw up your DNA. So that's a choice, right, if you're doing that. And last but not least, you want to go and get checked out. And I don't have a slide for that. But you would want to go get checked out, and you could either have your tissues checked, you could have your blood checked, you could have your DNA checked, and you want to be active in that checking to make sure if it's something physical you can check just with your own hands, and you want to look for that and get it checked out right away. And then the treatment right now is pretty harsh. You have to have surgery to have something cut out, or you expose your body to radiation or chemotherapy in order to kill off those cancer cells. But we said maybe in the future they'll establish new things like vaccines, like they have against HPV right now. Or you'll fix somebody's P53 gene, its assassin, so that they start shooting actual bullets, not blanks. That might be the future treatment. Or preventing angiogenesis. Right? That is it. I can throw up some questions if you want, or you can call it a day, whatever you would like. Questions? Okay. All right. And, oh, wait, let me also, can I give you some tips for the test? Okay. Some tips for the test. I would be very familiar and be able to draw 3 different things. A simplified drawing of 3 different things. DNA replication, transcription, and translation. I would be able to discuss my model, my strengths and weaknesses of my model, that I used. Remember for your HyperDoc how you had a model? I would be able to discuss that. I would understand the lac operon very well that we went over in prokaryotes. I would be able in eukaryotes to list all 5 levels of control. I would want to be able to differentiate between a cancer cell and a normal cell. What are some differences between them? And then I would want to identify the 2 genes that I'm concerned about and an example of each. So proto-oncogenes, becoming oncogenes, I'd want to talk about RAS. And tumor suppressor genes, I'd want to talk about P53. That make sense? There's some good tips. I know, because I wrote the test. Okay? All right. So back here, let's see. I don't think I have any questions on the top one. For some reason, I do not. But I have some here. Okay? Why don't we go in as how many hours of homework you have tonight? It's a number. Go in as a number. And I'm going to pause you for a minute. Okay, let's check your answers. Good. RNA polymerase binds to the promoter. Perfect. The repressor binds to the operator. The genes are located that code for the repressor. Regulator genes code for the repressor. Structural genes, for the 2 of you that picked that, that's whatever we're trying to ultimately control. In this case, it was the production of an enzyme to digest lactose. 4, lactose will bind to the repressor and inactivate it. And 5, the structural genes coded for those lactose digesting enzymes. All right, here, there's some other questions here. If you would like some more. And I'm going to go ahead and pause you. Okay, here we go. Getting introns removed is post-transcriptional control. Some of you said transcriptional control. In fact, 5 of you did. The making of the mRNA is transcriptional control. The modification after it's made, cutting out introns, cap tail, leaving a nucleus, is post-transcriptional. Okay, 3-dimensional folding is post-translational, not post-transcriptional. Transcription, I only have RNA. If I have a protein after post-translational, right, I have that protein. Number 3, you all got this. Bar bodies is chromatin structure. The ability to bind to a ribosome, you're doing translation. That's not transcription. Transcription occurs in the nucleus, right? Being out in the cytoplasm is where you do translation. Okay, and I think that's it. All right? Yay! Make good choices. Have good evenings. I'm glad you did this. Be proud. Have a piece of toast.